Good morning, everybody. Welcome to our Department of Psychiatry Grand Rounds. I'll introduce the speaker in just a minute. Just to remind you, at the end of the talk, there'll be questions, and then at 9:30, we're going to steal Danny across the hall for meeting with trainees. If you are a trainee here and haven't already signed up, there's still space available, and you get your own private time with Dr. Weinberger. We're really pleased to have Daniel with us today. He's the director and CEO of the Lieber Institute for Brain Development and professor of psychiatry, neurology, and neuroscience, and human genetics at Johns Hopkins Medical School. So most of you know that Dan was formerly the director of the Genes, Cognition, and Psychosis program at the NIMH Intramural Program. He attended medical school at UPenn, where he first met our chair, Dr. Grant. I'll let him tell you how that happened. He interned at Harvard General and did residencies in both psychiatry and neurology, the first at Harvard, the second at George Washington, and he's board certified in both. Dr. Weinberger's research focuses on brain and genetic mechanisms involved in the pathogenesis and treatment of neuropsychiatric disorders, especially schizophrenia. He's been instrumental in focusing research on the role of abnormal brain development as a risk factor for schizophrenia, and most recently identifying specific neural and molecular mechanisms for genetic risk for schizophrenia. His recent work is focused on the genetic and epigenetic regulation of expression in the human brain of genes associated with developmental disorders. His CV lists 665 peer-reviewed publications, 222 book chapters, and 11 books. Dan's the recipient of many honors and awards, and I'd be here all morning listing them. If I went through them all, I'll just mention a few. The Judah Silver Award from NAMI, the A.E. Bennett from SOBP, Joel Elks Award from ACNP, and a whole list of others, the NIH Directors Award, the Kemp Fund Award, and the Foundations Prize from APA, and the Lieber Prize from the Brain and Behavior Research Foundation. He's the past president of both the SOBP and the ACNP, and has been elected to the National Academy of Medicine. Today, his Grand Round presentation is entitled Genomic Insights about the Neurodevelopmental Origins of Schizophrenia. Please join me in welcoming our esteemed colleague, Dr. Dan Weinberger.
So, thanks, Neil. Good morning, everybody. This is where I come from. This is an ungodly hour to have a psychiatry grand rounds, but I'm glad to be here. It's really good to see a lot of friends that I've known for quite a while. Everybody looks terrific, and it's a joy to be here. Spent a lot of time in San Diego in the last couple of weeks, and I'm getting very familiar with it. So I'm sort of wondering how much more time I might be able to spend here in the future. As Neil mentioned, I'm going to tell you a little bit about our thinking, current thinking about the role of brain development, in particularly schizophrenia, but these are probably principles that I think apply to many developmental behavior disorders. It's pretty obvious to anybody that has children that there are many aspects of human temperament that are inborn from early in life, and it's led to the obvious question of whether there are developmental components to neuropsychiatric disorders. This has been a subject that has been of great interest to me for over 35 years, and it was about a little over 30 years ago I published a paper in which I suggested that if we really wanted to understand a disorder that we think of as occurring during young adulthood, that we call schizophrenia, we had to understand how the brain was built to establish a trajectory that would read out as this particular syndrome when people reached early adult life. And I also made the statement at that time, which, and the fundamental argument that I made was that there were early developmental deviations in a normal developmental trajectory, which interacted with many normal developmental events during early adult life that resulted in the expression of this phenotype. And I also suggested at that time, which I now feel even more strongly than I did then because we had very little evidence for this, that schizophrenia wasn't really a disease in the typical medical sense of the word, rather than a state of brain development that had a particular way of experiencing postnatal life. Since that time, there have been many, many studies looking at the early developmental histories of people who developed schizophrenia in early adulthood, and these studies have documented many deviations from normal developmental milestones that exist even in the first 6 months of life that are enriched in the life histories of people who develop schizophrenia as adulthood. And there are many, many studies of this. This is probably the most consistent epidemiological literature in schizophrenia, that early developmental antecedents are enriched in the life histories of people who manifest schizophrenia in early adulthood. But now that we have something much more about the egg and not the chicken, meaning these are all observational kinds of findings. We have genes now. And we can ask a question, does the genetic evidence for basic causative mechanisms of schizophrenia support this model? Because all of these observational phenomenological findings could be the chicken rather than the egg. Genes can only be the egg. They are the basic molecular starting points for these trajectories. So just to get us all on the same page, let me remind you what the current state of the art is in gene discovery related to the phenotype that we call schizophrenia. We've known for many, many decades that schizophrenia runs in families, and we've known from the pattern of inheritance in families, which is non-Mendelian, from the relative risk found in identical twins who are discordant for illness, that and from children adopted away at birth, that these conditions run in families, not because of the reasons that language or religion runs in families, but because of inheritance. And because we've been able to do direct genetic analysis now, we can identify specific regions of the human genome, specific mutations, specific chromosomal abnormalities that are associated with increased risk. So we know that by doing sequencing of specific nucleotides, so mostly exome sequencing, that about 1 out of every 200 patients with the diagnosis of schizophrenia have a rare loss of function mutation in a protein coding sequence of the genome that probably is a significant moderate risk factor for illness. This is more common in people with autism spectrum disorders. We also know, this is largely the work of Jonathan Sabat here, that patients with neuropsychiatric disorders have occasional cases where a large segment of the genome, somewhere in excess of 100,000 nucleotides, is either deleted or duplicated, and there's a gene dosage change within that region of the genome. These are called copy number variations. They occur in about 2% of cases that get the diagnosis of schizophrenia. They're much more common in autism, but they're also found in rare cases of ADHD, etc. The majority of cases with the diagnosis of schizophrenia seem to have their genetic origins based on common variations in the genome that all of us have some of. This is about 97% of cases with schizophrenia, and even the majority of cases with autism have disorder related to variations in the genome that are common in our species. And it's the combinations or the plurality of these common variants that seems to account for genomic risk. So this is just to get you on the same page for thinking about what the genes, the catalog of genetic risk factors are that have been identified so far. So we can ask a question about whether these genes that we think have something to do with risk are about building brains or about how brains function after they're built. And so we've done a number of studies trying to explore this question. And we asked a very simple question, which is, if they're about building a brain, they should be abundantly expressed in fetal life. And we could go so far as to hypothesize that they might even be expressed more abundantly during fetal life than in postnatal life. And if we found that they were more abundantly expressed during fetal life than postnatal life, it would support the idea that these had some particular meaning to the building of a brain. So what we did was, in the first study, we put together sets of genes that were identified from all these different strategies I just mentioned. So we picked all the genes associated with a group of what we think are developmental disorders, which include schizophrenia, autism spectrum disorder, intellectual disability, and syndromal neurodevelopmental disorders. These are profound neonatal encephalopathies that are present at birth, where there's absolutely no question that the brain during fetal life was on an abnormal developmental trajectory. We looked at other sets of genes associated with conditions that are less clearly developmental, particularly neurodegenerative disorders of late life. This is the entire set of genes from CNV regions, from common variant associations, and we put these together and asked a question of the distribution of expression of these sets of genes. Was the distribution of the expression greater during fetal life than postnatal life? In order to do this kind of a study, you need to have human brains. And you need to have human brains for people who have lived across the human lifespan, but also from fetal brains. And this is the tireless dedication over 30 years of the work of Tom Hyde and Joel Kleinman. The Lieber Institute for Brain Development now has over 3,000 human brains. We're collecting over 400 brains a year, including about 100 fetal brains. So we could ask this question. The first study that we reported on this question was based on an oligonucleotide array analysis. This was an array that's available if anybody wants to go to this Brain Cloud website, which was developed as a partnership between the Lieber Institute and NIMH, which is looking at expression of genes that are identified by a set of nucleotides that represent common transcripts in these genes. And this was basically the first study. This was work of a postdoc of mine, Rebecca Birnbaum, now at Mount Sinai. And this is the distribution of all genes expressed on this array, this oligonucleotide array. And if the distribution is displaced to the right, it's greater expression during fetal life. If it's displaced to the left, it's greater expression during adult life. As you can see, the distribution of about 17,000 genes that were represented on this oligonucleotide array showed a fairly even distribution. But now let's look at the two extremes of the lifespan time periods: syndromal neurodevelopmental disorders and neurodegenerative disorders. The syndromal neurodevelopmental disorders, which is represented by 33 genes, these genes are differentially expressed as a gene set during fetal life compared to postnatal life. In contrast, the genes associated with neurodevelopmental disorders, which are frontotemporal dementia, Alzheimer's disease, Parkinson's disease, etc., there were 59 genes in that gene set. They are displaced differentially during adult life. In fact, it was late adult age. So this at least establishes the poles and the feasibility of this strategy, that we can use this strategy to identify sets of genes that represent risk factors for early life developmental disorders and late life neurodegenerative disorders, and they segregate nicely in a lawful, expected way. And this is what happened with schizophrenia, with autism and intellectual disability. As gene sets, these all supported the notion that these were more likely to be expressed during fetal life than postnatal life, suggesting that they clearly are regulated during a time of life where brains are being built with disproportionate expression compared to time of life after the brains have been built. So that was a study with a low-resolution oligonucleotide array survey of transcripts in the brain. The litmus test of gene expression is measuring all expressed sequences in the brain using RNA sequencing. RNA sequencing, like DNA sequencing, reads the expressed nucleotides in the transcriptome, and RNA sequencing offers the opportunity to assay transcripts, not based on what you know about a transcript, but based on what you discover in assaying the expression of these transcripts. So, as you know, you have a genome which has interspersed non-coding regions called introns, which have to be spliced out to produce a transcript. So we can assemble, because all these read technologies for RNA sequencing, with very rare exceptions, are short read technologies. The average transcript is about 2,000 to 3,000 nucleotides, and the average read length that we get is about 300 base pairs. So we don't ever read through the entire transcript. We have to take these short segments and somehow assemble a transcript based on that. So we can take all the reads that map to the known physical coordinates of a gene and assemble genes that we then count to look at differential expression, We can count all the exons, just the coding sequences, by mapping reads to known boundaries, physical boundaries of exons. We can just say, let's look at any part of the genome that's expressed, even if it's not in our databases, even if it's not supposed to be coding. This would be called an expressed region. So here's a expressed region that's not part of a known transcript. We can assemble what are the known transcripts in the databases. So when you assemble a gene, you're theoretically assembling families of transcripts, but we can assemble individual transcripts. And lastly, we can assemble something that we call junctions, which are reads in the RNA sequencing that have nucleotides from a little bit of this exon and nucleotides from a little bit of that exon, with the spliced out intron missing. This junction identifies these two exons, and we can do this across the genome. When you do this, you discover a few surprises. So for example, look at this junction. This junction has nucleotides from this exon and nucleotides from that exon, meaning this exon is missing in that junction. This exon has been spliced out of that junction, and this identifies a novel isoform of a transcript. This is the way you identify transcripts that are not in your databases. And as I'll show you, this is a very common thing in the human brain. Most genes processed in the human brain do not look like they look in Ensemble or UCSC. They're much more complicated than that. The other reason to do RNA sequencing is to realize that because most of the common variants that I mentioned that account for risk in about 97% of cases with schizophrenia and the majority of cases with all neuropsychiatric disorders are non-coding. They don't affect the protein coding sequence. They don't affect the amino acid translation. So if the gene is a risk factor for the biology of illness and they don't affect the protein coding sequence, there's only one way they affect the biology of the gene, and that's affecting expression, processing, etc. And there are many mechanisms, and there's a whole lot of literature now trying to understand three-dimensional chromatin structure, microRNAs, many different effects at the molecular genomic level that translate into variable processing of gene expression. But whatever it is, it has to read out in the transcriptome. And the molecular pathology is not the 3D chromatin structure. That's a tactic. The molecular pathology of the cell biology is the transcript. So RNA sequencing is the gold standard for understanding the molecular pathology of genomic risk when you're dealing with non-coding variants. So the first study we did using RNA sequencing was to look at the normal developmental landscape of all expressed regions of the genome across 6 different developmental stages, starting with fetal life, immediately postnatally, early adulthood, midlife, and late life. And we developed a tool called the DER Finder tool, which is differentially expressed region finder, which uses single base resolution to look for any expressed region of the genome that varies across these 6 developmental stages. So here's an exon for SOX11, SOX11, and this is fetal life, this is immediately postnatal, and these are later ages. This particular exon is most abundantly expressed in fetal life. So we did this for 36 brains. We replicated it in an additional 36 brains, 6 for each brain region. This is in the prefrontal cortex. We identified 50,000 regions of the transcriptome that had differentially expressed sequences across these different age groups. We replicated that in a third public data set called BrainSpan, and we also showed that this was a characteristic of mammalian cortical development, that analogous regions of the transcriptome showed this pattern in mouse cortex, and also in pluripotent cells differentiated towards neuronal fates. They showed very similar patterns. So this is the overall summary of that work. This is the work of Andrew Jaffe, who's a lead data scientist in the Lieber Institute for Brain Development. 50,000 DERs, 40% of these map to non-exonic sequences, map to sequences that are not supposed to be expressed regions. Most of these DERs, including what were the intronic DERs, are actually mature RNAs. We compared cytosolic RNA expression and nuclear RNA expression in these samples and showed that these are not pre-RNAs that are not meant to ultimately be translated. The majority have highest expression in fetal life. And these genes overlap, at least the ones that are particularly expressed during fetal life, with what you expect such fetal regulated genes to be about: neurogenesis, signaling, development, etc. So here was the results of what was found across different transcriptional features in schizophrenia, which include all expressed regions, exon, intron, as well as intergenic expressed regions of the transcriptome. These were differentially expressed during fetal life in schizophrenia, also in intellectual disability and in syndromal neurodevelopmental disorders and autism. Alzheimer's disease and Parkinson's disease, we again showed a significant enrichment, differentially expressed regions, but now, once again, this was during late life and not during fetal life. So let me show you an example of why junction reads are so critical in understanding differential regulation of gene expression. So this is actually from a more recent paper. This is a gene. It doesn't matter what the gene is. And there's a family of transcripts. And the family of transcripts are identified by different exons, different junctions. So if you add up all the reads that map to these physical coordinates of this gene, which is the gene level analysis, which is 95% of every paper you read in literature, is a gene level analysis. You get a pattern like this across development. This is fetal life. Every dot is a human brain. Normal, these are all normal people, supposedly. This is immediately postnatal. This is childhood, adult life. There's very little evidence of a developmental pattern of gene expression. So if you did this study based on gene level analysis, which is what most people report, you would say this is a pretty stably expressed gene across development. But let's look at different transcripts here. So this junction right here, which has reads from this exon and this exon, and identifies a family of transcripts. There are 4 transcripts that have that junction. This junction shows a very clear developmental pattern. That is very low abundant during fetal life and gets much more abundant postnatally. These are the kinds of p-values that you see at a biological level in transcriptional regulation in human brain. They go so far beyond what we're used to seeing in our GWAS data, because these are biological readouts of genetic variation, very close to the molecular mechanism of the effect. But this junction, which identifies a unique transcript, which is only found in this transcript, having nucleotides from this exon and this inserted exon here, has a completely different developmental pattern. We call this an isoform switch. The same gene is encoding 2 isoforms that have developmentally opposite expression patterns. And of course, that explains the lack of seeing any clear pattern across development. But this is a very exquisitely regulated gene across human development. Turns out we've looked at these isoform switches with respect to schizophrenia. They are enriched genes having some transcriptional feature that switches from prenatal to postnatal life directionality encoded by the same gene are enriched in schizophrenia loci coming from the GWAS data, suggesting that something about genes associated with schizophrenia are particularly meaningful in terms of this isoform switch regulation problem. So I want to also make the point, this is from a recent paper, that when you try to identify within loci emerging from GWAS data, and I think most of you realize that GWAS significant p-value loci are not genes. They're low-resolution map points across the genome. And most of these loci have more than one gene that have been physically mapped to that region of the genome. Most of them have many genes mapped to that region of the genome. And it's impossible to know from no matter how many SNP genotypes you do and how much bioinformatics you do, exactly what genes or what isoforms of those genes account for the clinical association. You have to do more. You have to do some molecular biology to determine that. And this illustrates this. This is from the schizophrenia GWAS. This is the PGC2 GWAS. This is the whole National Human Genome Research Institute catalog of GWAS association across many diseases, showing that the EQTLs are about, just in one tissue now, looking at any transcriptional feature, which includes junctions, exons, etc., about 50% of them, just in prefrontal cortex alone, do associate with expression of a particular transcript feature. But if you ask how many of these loci have only one transcript in them, it's about 10%. 90% of them have many transcripts in them. About close to 10% are only associated with a transcript that was previously unannotated, meaning nobody had ever found it before or identified it before. And about half of them include, in those loci, include previously unannotated transcripts, which might conceivably be a culprit. So this is just to illustrate the complexity of using simply the visual imagery of where loci are, where genes are, to draw inferences. All of the data I've just shown you are publicly available to anybody that wants to look at it. This is in a website called eqtlbrainseq.org, which was a project that the Lieber Institute for Brain Development did with 7 drug companies to produce a public data set, a very detailed gene expression analysis across the human lifespan. This involves from fetal life to the 90s. There's about 800 human brains in these data sets. It includes differential expression of patients with schizophrenia and controls. There's also an EQTL browser, and it references the Common Mind data and several other GTEx and other data sets where you can look at similar EQTL analyses and developmental expression. We have DLPFC and hippocampus in this now. The caudate will be in very soon. So and it's pretty user-friendly. You don't need a lot of tutorials to be able to use these data. So the other thing we were very interested in understanding is what role early environmental influence might have on these differential gene expression profiles. And it's pretty clear that there's a lot happening environmentally during fetal life, and that the only way you change expression, splicing, processing of a gene from the same genomic sequence is to change the epigenetic state of that genomic sequence. So we did the exact same study now looking at a marker of epigenetic state, which is DNA methylation. This was using a low-resolution CPG chip, the Illumina 450K chip. But we identified this exactly the same study, but now instead of looking at gene expression, we looked at changing landscape of DNA methylation across the genome from fetal life through the human lifespan. We identified about half of these methylated cytosines that showed this differential pattern across development. And these are pretty abundantly associated with gene expression. This is the landscape of this. Significant enrichment of these CPGs that showed were highly methylated in fetal life compared to postnatal life were found within the Psychiatric Genetics Consortium GWAS study for schizophrenia. So the loci identified in the GWAS study for schizophrenia, that those loci showed this pattern of differential methylation in fetal compared to postnatal life. This was not seen with Alzheimer's or Parkinson's disease. We also, because we were looking at normal brain, we could look at the change in the epigenetic state of the genome around the time that people make the diagnosis of schizophrenia, which is between the ages of 18 and 25. This is obviously a tumultuous time of life. There's a lot happening environmentally. There are many changes in the epigenetic state of the genome during that time. So we'd identified these fetal epigenetic changes to be enriched for the loci associated with schizophrenia, the GWAS study. What about these epigenetic changes occurring around the time that we make this diagnosis? These were not enriched for PGC loci. There was no relationship between what was happening in the normal brain at the epigenetic level between age 18 and 25 and the regions of the genome that have been identified as risk factors for schizophrenia. The last thing we could say was, okay, what about the state of schizophrenia? So the average age of brains in the Lieber Institute brain repository is about 45, which means these people have been sick for 20 or 25 years with the diagnosis. They've had all kinds of medicines. Many of them have taken various drugs. They have poor medical care. They obviously have enormous psychological distress, or many complex environmental adversities that you would expect to create a marked epigenetic difference between patients and controls. So we said, okay, let's find the regions of the genome that show DNA methylation changes between patients with schizophrenia controls. And let's ask whether those regions that differentiate illness after 25 years when people die from wellness echo the findings about early development or echo the findings around the time that they were first diagnosed. Basically, what we found was there were about 2,000 slightly differentially methylated CPGs between patients with schizophrenia and controls. These were strongly enriched for something that happened to these people 45 years ago that left an enduring, lasting epigenetic mark on their genome. Despite all the vagaries of their life, these marks were still there 45 years later. Those things that happened around the time they were diagnosed are literally depleted in the DNA landscape, methylation landscape of these people. So these results, and I think it's important to remember, when you don't find anything, you haven't really proven anything. But these results suggest that risk factors for schizophrenia, both genetic and those environmental factors that leave a mark in adult brain, are principally related to early brain development and not to the tumultuous time of clinical diagnosis. One other thing that we observed in the methylation data, we've now done a big bisulfite sequencing data of the entire DNA methylation landscape across the genome and development. But at this time, we discovered that almost 2/3 of the GWAS-associated index SNPs, those that were GWAS significant 10 to the minus 8, were associated with the methylation state of the genome around that SNP. And these are spectacular p-values. I mean, this is 10 to the minus 92. This is only in a few hundred brains, because these non-coding variants affect the epigenetic state of the genome. This is their biological readout. And together, this suggests, which I think is going to be the story for all common variants related to psychiatric disorders, these are about how environmentally sensitive and responsive the genome of people are. These genomes, these epigenetic states are determined by sequence. The sequence sets the stage for what regions of the genome are modifiable by an environmental experience and what particular environmental experiences might matter to a particular genome. But I think this will be the rule as we get higher resolution maps of DNA methylation changes. So this is very interesting biology. But one thing pointed out in all of these studies is how little individual risk is explained by any of these findings. It's often said that the common variants have odds ratios less than 1.1. So if everybody has a probability of having schizophrenia, 1 out of 100, if you inherit one of these risk alleles by itself, that has a p-value of 10 to the minus 25 in the latest study of 100,000 people, it increases your risk not to 1%, but 1.1%, which is meaningless at the clinical level. So one of the very interesting, why are these risk effects of common variants so weak? And why is it, which many other people have made the observation, that the human genome did not evolve to validate the DSM-5 criteria of psychiatric illness? These variants bleed onto many other disorders. So I can't help but not mention a very provocative and thoughtful hypothesis to explain some of the pleiotropy that's been described with these common variants and the small effect size, which is a hypothesis that Jonathan Pritchard at Stanford published now a year and a half ago, suggesting that the reason these risk variants are of such small effect is because basically the principal findings from GWAS is that any gene expressed in a cell that matters to your illness will contribute to risk for that illness, because they're not there if they didn't matter to the cell. Only genes that matter to the biology of a cell are expressed in that cell. And his argument was, while there may be some genes that matter more than others, which will be very difficult to distinguish from the peripheral genes that matter simply because they're there, is because cells operate based on many homeostatic interacting networks. And even though a gene might not be a critical, what he would call core gene, they're in related networks that interact with those critical networks. And if you genotype 1 million people or 5 million people, you'll discover that every gene expressed in a neuron will play a tiny role in risk across that diverse population. And we will probably in 10 years have in our gene quivers common variants that contribute risk fractions of fractions. This is a provocative hypothesis. It's interesting biology, it's interesting theory, but what else is missing? So the epigenetic data argue very strongly that what's also missing, and this is true for every complex medical disorder, is the environment. There's no question that the environment is a factor, not just in psychiatric disorders, but in all common medical disorders. Problem with the environment is it's very nebulous. Genes are bounded biological entities which are fun to study because you can define them, define the rules by which you observe them, you can quantify them, you can play tricks on them. The environment's much more difficult. And it's probably infinite. Genes are finite, but the environment's infinite. So how do we do this? So the three things we know about schizophrenia, for sure. The first is that it is a polygenic architecture. We didn't need GWAS studies to know this. We knew this from linkage studies. We knew this from family segregation studies, that the pattern of relative risk among family members is not consistent with Mendelian inheritance. It's been clear for a long time that polygenic architecture characterizes these common disorders. We've also known before we did gene expression studies in fetal brain that there was something about the early childhood environment that mattered to risk. And the data I love to cite because no one ever talks about it, but it was an elephant in the room in thinking about relative risk data in families. If you look at dizygotic twins and siblings from unrelated pregnancies and their relative risk to share illness if one of them had schizophrenia, dizygotic twins and siblings from unrelated pregnancies are equally genetically similar to their sibling. Dizygotic twins share the same, on average, alleles across the genome as do siblings from unrelated pregnancies. The only difference between dizygotic twins and siblings from unrelated pregnancies is dizygotic twins share the same uterus. And the frequency of the concordance in dizygotic twins is about twice what it is in siblings of unrelated pregnancy. This has never been explained. There have also been many studies showing that within sib pairs, there's a within monozygotic twins, there's a disproportionate likelihood that if one of the two twins is the twin that develops schizophrenia, it will be the smaller twin and the second-born twin. So, and the third thing we've known is that all developmental behavior disorders, schizophrenia, autism, dyslexia, ADHD, Tourette's syndrome, they're 2 to 4 times more common in males than females. We've never had an understanding of this. There have been many theories about it. So we decided to look for gene bioenvironmental interactions and see if we could have a better understanding of how genetic risk translates into illness if we had some hold on the environment. And the environment we picked, which is the environment that has been most consistently implicated in early developmental risk for all these developmental behavior disorders, is complicated pregnancies. Complicated pregnancies happen in 15 to 20% of pregnancies. It's not a rare event. It's a much more common event than is the differential allele frequency in a common variant between a patient and control. So for genomic risk, we picked what's called the polygene risk score because nobody gets sick with one common variant or another common variant. It's the complement of genetic variation across your genome, which has been calculated using a very simplistic additive sum of all risk-associated alleles across your genome that are the same risk-associated alleles that were found in the latest GWAS study. And as shown in this original description of this, if you're in the highest decile of risk of these poly risk scores compared to the lowest decile, there's about a 10 to 15-fold greater likelihood that you'll be a patient with schizophrenia here than here. This is comparable, by the way, to CNV risk-associated, more penetrant variants. So this has been a very popular way of having a sort of black box assessment of overall genomic risk of an individual for illness. For environmental risk, we use complicated pregnancies. complicated pregnancy has been found in many studies to be risk factors for ADHD, for schizophrenia, where the odds ratios are increased 2 to 4-fold, for autism, many, many studies. For Tourette syndrome, we actually reported this about over 20 years ago now, showing that if you look at identical twins, 10 years of age, who vary in tic severity, that almost the entire variance in tic severity when they're 10 years of age, and they both have Tourette syndrome, was explained by different APGAR scores at birth. So that was the basic model. And so here we're looking at polygene risk scores. And polygene risk scores, as you know, can be defined by adding up all the alleles from the GWAS study, all the loci, the index SNPs from the GWAS study, at different p-values of association. So you can have a polygene risk score based only on the SNPs that were GWAS significant at 10 to the minus 8. You can have a polygene risk score adding up all the SNPs that were 10 to the minus 6, 10 to the minus 4, etc. But this is now looking at just what's called the polygene risk score 1, which is based on the index SNPs, the 108 loci SNPs that were GWAS significant. And what we find in a group of patients with schizophrenia and a group of controls, what every group finds now. As a group, patients with schizophrenia have higher schizophrenia-associated polygene risk scores than do controls. And this is a highly significant difference. But now we say, but what happens when we look at the same analysis in the context of a serious early life complication? And we were very shocked to discover that in a discovery sample, the difference in polygene risk scores was only found in the sample that had history of early life complication. And there was a highly significant interaction between the genetic risk and environmental risk. And actually, in the group that had no early life complications, patients with schizophrenia, there was no difference in this. This interaction, by the way, is very interesting because it not only is made by the patients with ELCs having higher polygene risk scores, but the controls with ELCs have lower polygene risk scores, which would be consistent with the idea that another risk factor to increase the probability of someone ending up in one bin or the other has to be counteracted by a resilience factor. So this was a very rarefied result that we didn't really imagine could be replicated, but as it turned out, in the Psychiatric Genomics Consortium, there were 2 other samples that had both detailed obstetrical information and GWAS data, and they both showed essentially the same thing. That there was an interaction between polygene risk score and serious obstetric complications, and individuals who had the serious obstetric complications had higher polygene risk scores than those that didn't. Let me illustrate for you. This is the analysis of the whole sample. This is the interaction of the 2. Let me illustrate how much of a difference this makes. So here is the classic GWAS data for risk. This is a measure, a modeling of risk in the population, how much risk is accounted for by genomic risk. So it's about 3 to 4% is accounted for by this measure of genomic risk called the polygene risk score 1. But if we look at the samples with early life complications and without, there's about a 5 to 7-fold difference in risk prediction if you've had an early life complication and the same polygene risk score than if you don't. And this was true for both the polygene risk scores calculated from the index GWAS significant SNP loci SNPs, the 10 to the minus 8 SNPs, and the 10 to the minus 6 SNPs. Here, you're looking at the actual ratio of change. So at the polygene risk score 1 level, there's about a 7-fold increase in risk compared to having an early life complication or not. This is only found for these 2 loci-derived polygene risk scores. When we go to 10 to the minus 4 and less significant, where it is generally thought most of the schizophrenia genes are found, but the sample sizes aren't large enough yet for them to reach the rarefied level of significance, there's absolutely no relationship whatsoever between having an early life complication and not. There's something about genes in these loci that interact with an early life complication. And we suggested in this paper, which was out last June, we suggested that The reason the 10 to the minus 8 loci are the 10 to the minus 8 loci is because they interact with a common environmental event that allows them to achieve a certain level of statistical significance that they would not achieve without that. But what is it about the genes in these loci? I also should point out that these effects only are seen when there's a serious obstetric complication that has been well documented in the obstetrical literature to be a physiological risk to the fetus. So these are scores. This is a well-validated score for a degree of obstetrical risk to the fetus. Scores of 4 and above are scores that are thought to represent a significant obstetrical risk. These are things like preeclampsia, intrauterine growth restriction, rupture of membranes without labor, infection of amniotic membranes, etc. At mild obstetric complications, nuchal cord, delayed labor, forceps delivery, there's no interaction. This is about something that's happening to the fetus. So the question is, this is just to show that we did this also in 2 samples where we didn't have controls, but included a Japanese sample where the polygene risk score is much less of a risk factor because of the ancestral haplotype variants in Asian samples, but we still found the same thing. And this was the work of my 2 postdocs, Gianluca and Giovanna. Okay, what's going on in the uterus? And what is it about these genes that make them more susceptible? What's happening in the uterus? And this made it impossible not to ask the question, what is happening in the placenta? Because the placenta is, in essence, a fetal organ. The only part of the placenta that comes from the maternal genome is the basal plate that lines the intrauterine membrane. All the rest of the placenta comes from the fetus, just like the lungs, the heart, the brain. It's another fetal organ. It has the fetal genome. It has these genes associated with schizophrenia that are risk factors in the fetus, and the mother does not have the full complement of these genes. What's happening in the placenta? So we had the opportunity now to do RNA sequencing analysis on multiple placentae, multiple components of the placenta. And what we're looking at here is the differential expression in the placenta of the genes in the loci that are mapped to the loci that show interaction with obstetric complication compared to all the other schizophrenia genes in the loci that don't show the interaction with obstetric complications. And what you see here is in all these different compartments of the placenta, these are dramatically upregulated genes. The genes in those two gene set loci, the PRS1 and the PRS2, the ones that interact with obstetric complication, in contrast to all the other PRS loci genes, where most of the schizophrenia genes are thought to be, but that don't interact with obstetric complications. The ones that do are much more upregulated in placenta, in normal placenta at term. But we also had the opportunity to look at some placentas that were from complicated pregnancies, preeclampsia, intrauterine growth restriction, and now we could look at how the same genes in the same loci compared to the other schizophrenia gene loci are dynamically regulated in placenta from complicated pregnancies. And in every one of these data sets, this is looking at, again, the distribution in red of the whole set of genes in these loci. They're all differentially regulated, upregulated, dynamically regulated in placentae from complicated pregnancies. These were 8 different placental data sets from different, and this included a study of invasive versus non-invasive cytotrophoblasts. These genes are not only very abundantly expressed in the placenta, but in placentas that are under duress and stress during complicated pregnancies, they seem to be upregulated even more. We did a number of sensitivity analyses to look at many other organs under distress. We looked at many fetal organs. None of these show this pattern. None of them show this pattern. This is unique to the placenta. This is not a manifestation of these schizophrenia genes happening to be sensitive to anything going on in a human body. They're very sensitive to something going on in the placenta. We also were able to go back from the data in the placenta to ask if these observations actually explain the interaction with complicated pregnancy risk for schizophrenia by deriving a new polygene risk score. So the polygene risk score we derived was based on the SNPs that marked the loci having genes dynamically regulated in placenta from complicated pregnancies compared to the other loci gene polygene risk scores. And what you see here is, we call this the placental polygene risk score 1 and 2. These are the genes these are the summation of all SNPs in loci in the genome, marking regions where there are genes dynamically regulated in the placenta, and the non-plaque ones are the same loci, but not with placental-related genes. And you can see there's no interaction here whatsoever. This interaction is explained by the genes expressed in the placenta. We also looked at what these genes were about biologically. We did a number of so-called bioinformatic analyses and silico analyses to ask, based on what's known about the biological processes of genes, how do these genes in the placenta look like genes that have previously been associated with schizophrenia? This is one of a number of snapshots, but the genes that are not in the placenta, but are in GWAS loci for schizophrenia, we call them non-plaque genes. These are about synaptic function, calcium signaling, etc. The genes that are in the placenta, in the schizophrenia risk loci, interacting with obstetric complications, these are about a completely orthogonal biology. They have nothing to do with synapses. They have nothing to do with calcium signaling. They're about basic cellular responses to stress and to hypoxia. So how do these placental genes influence placentation? What do they have to do with a placenta not functioning like a normal placenta? Because obviously, the placenta is the critical organ for the development of the fetus. The fetus goes nowhere without its placenta. So animal studies suggest that many placental stresses invoke an inflammatory and an immune response to the placenta. It's very important to realize the placenta, I've learned a lot about this from our obstetric colleagues. I knew nothing about the placenta until about 2 years ago. Placenta is an alien tissue in the body of the mother. It is principally derived from an alien genome. It is highly invasive. It is a tumor. It is an alien tumor. It grows incredibly from a few cells to 1.5 pounds in 9 months. It is highly invasive. It is a much more rapidly invasive growing tumor than any form of cancer. The only thing that differentiates it from a non-metastatic cancer is it sort of comes out at the end of the 9 months. But animal studies suggest that no matter what you do to a pregnant mother, that it poses a physiological stress, which can be psychological stress, changing protein diets, changing exercise, changing oxygenation, that there is an immune response initiated by the placenta. And this is a sign of the degree to which the placenta is dealing with some sort of stress. So we asked the question of whether or not a group of 560 genes related to immune responses and inflammatory reactions were tracking in their expression, the expression of these genes associated with schizophrenia in the placenta, and they are highly abundantly co-expressed. These schizophrenia genes are not immune genes, at least the ones in these loci. They are not inflammatory genes. They are marking the state of stress of the placenta as reflected in this immune response. So we think this has something to do with how they are engaged in this process of a placenta under stress. The last point I wanted to make was that we've never understood this question of male preponderance of disorders like schizophrenia, but animal studies, this is largely the work of Tracy Bale, who was at the University of Pennsylvania. She's now at University of Maryland, but many studies have shown this in animals, that all these stresses to pregnant dads tend to have a greater effect on the offspring in many developmental measures if the offspring is a male than if it's a female. There have been many hypotheses about the second X chromosome having some resilience effect in placentas, but this is poorly understood. But we asked whether there's a link between the greater incidence of developmental disorders in males, if this has something to do with how these genes are regulated the placenta. And basically, these are the data. We looked at the genes expressed in the placental placenta of complicated pregnancies if the offspring is a male compared to if the offspring is a female. And the bottom line is, if the offspring is a male, the genes in the GWAS loci, which is 1 and 2, that interact, that are dynamically regulated in placentas from complicated pregnancies, that identify SNPs, that interact in polygene risk scores for risk for schizophrenia, are much more abundantly expressed in males, in the placenta of males than the placenta of females. And this may be a factor in the greater preponderance of schizophrenia and many other conditions in males. This is a story fundamentally about schizophrenia. We're working on this in several other developmental disorders. It's hard for me to imagine that there won't be some analogous stories emerging from those as well. So these are the people whose work I've shown you, which includes our group from the Lieber Institute for Brain Development and outside collaborators who have contributed to this work. And the take-home message is genes associated with developmental disorders are preferentially expressed during fetal life. It's not to argue they don't play a role postnatally, but they're clearly playing a very strong role in setting up the early developmental trajectory from fetal life. GWAS loci enriched for genes showing transcriptional and epigenetic changes related to early fetal life and not for changes around the time of clinical diagnosis. The most significant genetic variants detected by current GWAS contribute to risk of schizophrenia, at least partly by converging on a developmental trajectory sensitive to intrauterine and perinatal adversity and linked with abnormal placentation. Gene-environment interactions influencing placental biology may account for the higher incidence of schizophrenia in males compared to females. Preserving prenatal health may represent a primary form of prevention of schizophrenia and maybe other developmental behavior disorders, especially in male individuals with high placental genetic risk. If you think about how modern medicine treats complicated pregnancies, it's principally with bed rest. This is not a very sophisticated 21st century treatment. And there have to be better ways to improve the resilience of the placenta under what is a 9-month period of extraordinary stress than the current technologies. Many of these data, the content of this talk is in a paper that Rebecca and I put in Nature Neuroscience about a year ago, a little over a year ago. Thank you for your attention. I look forward to any comments.
Danny, that's fantastic. I think this is exactly the purpose of Grand Rounds, is to get everybody to come in, think they're going to learn about one part of science, and then have their brain stretched and thinking about an entirely different part of biology here. And you've made a compelling case for why we should do that. I'd like to open up the floor here for questions from the audience.
Yes, very elegant work. As a chronobiologist, I can't help thinking, schizophrenia is part of timing disturbance. At least you have to be in the right place at the right time. I wanted to ask about a couple of roles of prenatal life in women who are pregnant who have sleep apnea. That can cause vasoconstriction to the uterine arterioles. I think Neil Richton's been doing some work on that. And I wonder about the role of that. And then Robert Friedman's work on choline and methylation. And Tim Oberlander from Vancouver has been finding that a little bit of stress is actually good for the fetus, and it kicks in the resiliency genes. And I'm wondering if that plays.
I mean, if somebody said to me, this is a new world of obstetric medicine. Obviously, there's been so little really done in a very deep way to understand what are the boundaries of placental health and what are the factors that influence placental health. And, the placenta is not going to be different than the brain in the sense that there are going to be genetic risk factors, there are going to be resilient factors, there are going to be environmental risk factors and resilient factors. It's a very complicated organ, by the way. If you look at the placenta compared to the placenta of other primates, the human placenta is much more complicated. There are many more cell types. This is an extremely complicated epigenetic landscape because this thing is changing profoundly over 9 months. And the epigenetic landscape of the placenta in different compartments, different cells are changing dramatically. Placenta makes a number of critical molecules for fetal development. I know people probably haven't tracked this very carefully, but for example, Pat Levitt published in Nature probably 10 years ago, that serotonin is not made, even though there are serotonin receptors in the fetal brain, it's not made till the 3rd trimester of fetal life. It's all made, serotonin comes from the placenta. And the placenta has this humoral serotonergic, bath of the fetus to keep these receptors active. Allopregnanolone, which is critical for GABA receptor maturation. The enzyme that makes allopregnanolone is in the placenta, it's not in the fetus. There are many examples of this that are of limited investigation. But, I think this is work that needs to, one of the scientists at the Lieber Institute for Brain, actually came from the Salk, is building human placentas in a dish from pluripotent stem cells. And they are very easy to make in contrast to a lot of neuronal models, because if you think about it, I mean, by the 5th division, you start making trophoblasts. And so it takes about 6 days to make a monolayer of placental cells of epitrophoblasts. And then she's made these 3-dimensional models that have villi sticking out of the plane of the, and they respond remarkably well to environmental stimuli of various types. So, I mean, I think this is going to be a new world of obstetric medicine. I think the other thing, I gave a talk at the University of Colorado about a month ago, so, to the pediatrics department. And the chief pediatric came up to me afterwards and he said, I think we have to start thinking about pediatrics as beginning before birth. Which is, by the way, long overdue that they would think that way. So, yeah, I mean, I don't know the answer to any of those specific points, but I think, it's something that needs to be.
Danny.
Beautiful talk, your usual tour de force. I just have a quick question about causality, and that is your hypothesis is that the schizophrenia genes, or a portion of them, make the placenta vulnerable to external events. But could it be the other way around, and that this subset of schizophrenia genes basically cause the labor events rather than?
So that's a very good question. Yeah, and I don't know that we can exclude that possibility. But I'll tell you what we tried to do, but we were very underpowered to do this. So we looked in the maternal genome for whether or not their polygene risk was predicted complicated pregnancies. We couldn't find any evidence of that at all. I mean, the p-value for that was 0.9. We did find a very weak trend, 0.15. Now, this was only like 250 families, so it's a very underpowered thing. 0.15 trend for the paternal genome to be associated with complicated pregnancies. This was more interesting than just this curious, likely to be not true finding, because animal studies have suggested that there's differential imprinting of the paternal genome in the placenta. And so, which is ironic, because obviously the father has nothing to do, with the exception of whatever psychological stress he might be causing the mother, which could be quite significant, by the way. All right, but this was in the genome. This was all genomic risk. So, again, we can't rule that out.
Thanks for a brilliant presentation, very thought-provoking. A couple of questions. One is, what is the role of fathers? There is some suggestion that the older age of the father increases the risk of schizophrenia. I know it has not been replicated. The second question is, if you look at the age of onset, typically below 30, it is men outnumber women, but after 30, since Kreplin's time, it has been known that women outnumber men for a later onset. So do you think that's a different type of schizophrenia in women with later onset?
So, I mean, there's no question that age of both parents, and this is a kind of, I think, smoky literature. There's no smoking gun in this story of age and its relationship to risk. There's clearly at the epidemiological level an association. And there's clearly an association both to maternal risk and to paternal risk. Paternal age, maternal age, maternal age. I think there's controversy about which one is predominant. And there's no question that there is an accumulation of mutations in sperm of older fathers, just like there's an accumulation of aneuploidies eggs of older mothers. But there's also a very compelling data set suggesting that what is critical for the association is the age at which the father has their first child, not the age at which they have a schizophrenic child. And these are several large studies from Denmark that has looked at this. We actually reported in Molecular Psychiatry 5 years ago a study in a large sample of families where we argued, okay, if there should be a birth order effect, same thing in autism, there should be a birth order effect in large families, that schizophrenic or autistic children should be the last born children in the family. They should tend to being the last born children in large families because those are the oldest father. The literature has never supported that. In fact, in autism, it tends to be the firstborn. And in schizophrenia, there's no birth order effect that's ever been identified. We showed in our sample that there was absolutely no birth order effect unless there was a complicated pregnancy. But the studies from Denmark, which are in the millions of people, have shown that there's clearly an age association with the father. But it's explained by the age at which they have their first child, not by the age at which they have the child with schizophrenia, which they have argued does not support that that age effect is based on accumulating mutations. It's based probably on some other factor, whether it be epigenetic age effects or the fact, which, that people who have their first child at age 50 or have their first child at age 45 are genomically biased in certain ways that may also overlap with some of the genomic biases for schizophrenia traits. So I think it's still a murky story what the causative relationship is.
We've got a quadrant bias here, so Abe, I'm going to come back to you. Is there any questions?
Thanks for a wonderful talk. Could you share your perspectives on next generation of treatments for schizophrenia?
So I have to warn everybody. There's a paper of mine coming out in the American Journal of Psychiatry in January called Thinking About Schizophrenia in an Era of Genomic Medicine. I'm talking a little bit about treatments. So, but I am very optimistic about treatments. Not just because I read John Kelso's papers, but also because I like to say that there is no treatment in psychiatry, whether it be talk treatment, drug treatment, electricity treatment, magnetization treatment. So the entire spectrum of psychiatric treatments, by the way, a little bit lacking to begin with. But there's no treatment that we use that was based on an understanding of causation or pathogenesis. So having some clues to causation and pathogenesis has to be a better strategy for finding new treatments. And I think as the field gets, which is getting very rapidly more sophisticated in ways to develop experimental models that could explore treatment mechanisms, new treatment mechanisms, I think I would just be surprised if in the next 10 years there are not several new targets identified from these new insights that have great potential to be. And they may be more personalized. Obviously, there's a lot of enthusiasm for personalizing treatment. I think that's a hope. But I mean, the problem with personalized medicine is that it's hard to know exactly which persons it applies to. I'm very optimistic about this. I mean, I think we have to have a better foothold now than we ever had in developing treatments for people. I mean, one of the it always bugs me, how much we see all this genetic overlap across these diagnoses. But we really don't see, despite this sort of loose discussion of this, we see remarkable segregation of treatment response, at least for the cardinal treatments. So, lithium is not an antipsychotic drug. It just isn't. It doesn't touch schizophrenia. Clozapine does not touch ADHD. It's terrible for ADHD. You don't give people with ADHD clozapine, and you don't give patients with schizophrenia stimulants. These are not therapeutic agents. That's not to say you can't improve some features of them, which are probably not schizophrenia unique features, but they're not antipsychotic. Stimulants are not antipsychotic. Lithium is not antipsychotic. ADHD is not attention deficit hyperactivity drug. You know, and so these clinical facts, you know, suggest that we have to have a more granular understanding of how these genes map onto different phenotypic features. I think we get this sort of complacent haze that goes over the field when we see all this overlap of genes, which are always very small effect sizes anyway. They're statistically significant because these huge sample sizes are involved. But there's obviously, it's obviously not characterizing these clinical phenotypes, I think, the way we would like it to. But nevertheless, you know, we always hear the argument that, well, you know, how is any one gene, even when you think you know the gene or you know the transcript, going to be a target when it only explains 1.1 odds of risk? But I think we're all operating on the assumption that these interacting networks, that you can reset this whole intracellular landscape by creating some sort of a perturbation in a particular node of these networks that will have downstream and upstream ramifications. Look, the bottom line is the drugs we use, which are not based on any of these clues to causation of pathogenesis, they do work to a certain degree. So you obviously can target a specific serotonin transporter, specific dopamine receptor, specific molecule, and get therapeutic benefit. So I don't think I think we shouldn't get too discouraged by the idea that any individual gene might not be a reasonable target because it explains such a little clinical effect. At a biological level, it could still have an important therapeutic effect.
Yeah. So you said that you didn't know much about the placenta years ago, and so I'm, like most people in the audience, 2 years and a day behind you on that. But I was struck that there were these placental expressed genes that seemed to be very important. And I wondered, placenta must have different tissue types, you briefly alluded to. I wondered if you're working with mature 9-month placenta to get most of this data, because that's what's available generally. Yeah.
Most of these data, not all of them, but most of them was from term placentas.
So you should be able to do single-cell RNA-seq and maybe figure out if there's a subset of cells that one may become more or less abundant when you have these complications, and that may drive the expression differences, that what you're really seeing is a cell type that became more abundant, and it's the cell type that expresses those genes.
Yeah.
And or you may be able to get a more nuanced understanding of where the action is in the placenta. Do you have anything like that, or is that,
no, so
I'm
that's
sure you're going there if you don't.
That's a to-be-done project. I mean, the problem, so we did look at multiple tissue compartments in the placenta, and generally, across these multiple tissue compartments, as a homogeneous tissue compartment, they generally show the same patterns, generally. We had very few placentas from early pregnancies. Right. Ironically, in the few placentas we had from early pregnancies, the differential gene effects were even greater. So we're now doing a study with first trimester placentas, which come from elective terminations of pregnancies for the most part. So they're, again, mostly normal, but that'll be, I think, a little bit easier study to do. You know, we assume, and there have been some epigenetic studies. There were some studies in autism looking at the epigenetic landscape of the placenta in autism, claiming that there were discrete epigenetic changes. This is going to be a very complicated epigenetic story because there's so much happening over a relatively short period of time. The placenta grows, you know, when you think about growing from 2 cells to a pound of tissue in 9 months, that's extraordinary. And this invasiveness of the placenta is amazing because it has to build this incredible vasculature to support this fetus, which is growing to be 9 pounds, 8 pounds. It's really quite a remarkable
pretty good.
organ.
I have a 100-pound dog that's only a year old, but I take your point.
Okay. You should do some single cell epigenetics on that dog.
Thanks for this elegant finding. I mean, what I see is since you have such a great amount of data to show that early life is really like a huge contributor in this kind of psychiatric.
Hold that a little closer.
That your like landmark finding that all the early stages is playing like a more than 99% for the effect of this schizophrenia and other psychiatric disorders. So how do you see the recent development in disease modeling, especially with IPS and organoids, which mimics the early stages, and even the more mature neurons are only like at an early fetal stage of developing neurons? And so how do you see those, the recent developments in terms of the findings where your earlier studies and now recent developments can provide some mechanistic influence? What are your thoughts on those?
So, I mean, I'm not a, I'm not a stem cell scientist, but I do sort of a voyeur of this field. So I think, it's a very exciting area of research, obviously, because they're human genomes. And they're living human cells, which make the unique model to study. The bad news about doing genetics is two main bad news stories. One is that you're studying human history, and you find things out about human history you may not want to know. And the second thing is you have to do cell biology. It's inevitable because these genes are about the biology of cells. I mean, genes don't know about hallucinations and delusions and panic attacks and depression. They know about the molecular biology of a cell. That's all they know. That's their universe. So you have to do cell biology. And obviously, we have a very rich, and I still believe very important, tradition of building models in animals to test specific hypotheses and specific biological and neuroscience systems, which are well worked out in animals, and there's tremendous conservation. But if you want to understand genetic variants in human genome and how they affect the trajectory of brain development, you need to start with human cells. So this is extremely, I think, just, it's a landmark development in basic science. And this field is exploding in technology, in sophistication. What started out 2 or 3 years ago as these monolayer models that had very limited differentiation into neuronal systems are now three-dimensional models that become very sophisticated. They can live for years. They can develop three-dimensional architectures. There was just a paper published that some of these little brain organoids have EEG profiles that have characteristics of child brain EEG patterns. Now, this is probably just the nature of how cells talk to each other, that they develop certain frequencies of activity that are recorded in EEGs. These are not brains. I would, by the way, if people did not get this, and I'll send it, you must have gotten it, our first issue of Neurodevelopments. So the Lieber Institute of Developmental has a newsletter, which is a quarterly newsletter. The first issue, I hope all of you got it. If you didn't, go to our website. You can subscribe to it. It's a very nice newsletter about neurodevelopmental issues related to clinical behavior. And the first issue was about organoids. And it was about what is the real value of organoids? What's the state of progress? It has a who's who editorial board of the principal scientists in developmental neurobiology today around the world. And it's meant to help us understand where these fields are going, why they might have relevance, what are the stumbling blocks, what are the challenges. It's very, very, lay-oriented, but this serious discussion. The editorial board has a discussion about certain topics. So we highlight 2 papers that are seminal papers in the field related to early brain development and clinical readout. And then we have a very sophisticated editorial board commenting on this. So I would strongly urge you to look at it. But this first issue, which was out about a month ago, was about organoids and what are the issues, how good are they, what aren't they, where will they go? I think, this is going to be a very dramatic area of science. I hate to overdo John Kelso, but their study using a model of lithium response and identifying a physiological characteristic of lithium responsive derived neurons from skin cells of people who respond to lithium and don't is a beautiful illustration of how you can use this model to learn something that you couldn't possibly begin to approach in any other way. Yeah. It's also when people talk about these models are about early brain development, it's important to remember that the programs that build synapses, the molecular programs driven by gene programs that build synapses, are for the most part the same programs that maintain the synapse. And they're the same programs that watch over the regression of synapses. So these programs keep getting reutilized over and over again in various different ways, slightly modified, slightly modulated. But it's not that far-fetched to imagine that even these basic models in very early neuronal systems will identify clues for how you can, rescue some problem that might also exist in a much more mature neural system.
Again, last question. So has to be very brief. As you know. So I'm always fascinated by your talk. And in talking with you, you are not talking about a theoretical genetic basis, but you also talk about treatment, which is very important. I just like to know what the future of immunotherapy for schizophrenia. I'd like to know your perspective on this matter.
First of all, Ming, if it's interesting, I'm very flattered that you find it of interest listening to me, because I always consider myself one of your students.
No.
So I give you credit for most of what you might find of interest. I think it's a controversial subject. Yes. I think the data, certainly the data in the periphery, in mood disorders, is impossible to dismiss. It's there. And I think the data in the brain in depression, where there's evidence of activation of immune pathways in the brain of people with depression, is more consistent than it is in schizophrenia. I would argue, and again, I have to defer to John. I don't know, I don't know why this has come up repeatedly, but you're much closer to this than I am. I think the big challenge in the immunological world of mental illness, and of course, the whole immunological question is sweeping through all of medicine now. And there's no question that many pathological processes engage the immune system and inflammatory responses. I think the big challenge for us is what's epiphenomenon and what's primary causation. I remember when I was at NIMH, probably in the 1980s, David Rubinow, I remember this, was doing these interferon studies. He was running the consultation liaison service at NIH. And I remember having lunch with David. He said to me, I've seen all these people getting interferon therapy, and they get this very severe depression. And he was very struck by that. And so there's no question that these inflammatory immune signaling molecules in the peripheral blood, some of them get into the brain, and they may have mood-altering effects from the periphery. That's one issue. The other issue is the brain itself expressing an immune response. I think those are two separate questions, but I still think the fundamental question are the immune markers, which are clearly found in the blood of many psychiatric patients, are they somehow a causative factor, or they are passenger to the stress? If you don't sleep for 24 hours, your C-reactive protein is up, your IL-6 is up. Stress causes activation of many of these immune responses. There have been hypotheses that that's a adaptive response, that you're meant to have this happen evolutionarily, because when you're stressed as a caveman, you need to be able to resist the infections or the being bitten by something, that you don't die of infection when you're being bitten by a snake or something like that. But chronic stress may cause a differentially adverse, non-adaptive response. I think these are interesting hypotheses. What's always worried me, Ming, is that when you give somebody aspirin, C-reactive protein goes down, IL-6 goes down. Many of these immunological factors go down. I'm not aware of aspirin being a therapeutic for any psychiatric disorder. And so I would like to see that, to have more confidence. Lithium was discovered by accident. Chlorpromazine was discovered by accident. And the accident was somebody said, these people are really better on this. I mean, how many people take non-inflammatory, non-steroidal anti-inflammatory drugs? with schizophrenia and depression. These drugs are taken by half the population. Some doctor would have noticed that when they were on their non-steroidal anti-inflammatory drugs, their ibuprofen, they weren't as depressed. So I hold out some note of skepticism on this, even though I have absolutely no doubt about the validity of the findings. But I think the jury is still out on how much targeting these immune responses will be therapeutic. I don't know if you, John, you may have a different perspective on this. I don't know.
Dan, thank you for the talk, for the discussion afterwards. I think I'll just say that paradigm shifts in any field require people to kind of reach outside that field. Others, this discussion of placental biology is just one example about how that might happen and what we might have to look forward to in our field. And for that, I'd like us all to thank you this morning for your talk. We do have one tradition at UCSD, as everyone here in the audience knows. Dan, you've spent so much time here, you earned a hat. So thank you. Thank you all. Trainees, your meeting with Dr. Weinberger will be